hard acts to follow, but I think we'll have Ron, Bartek, and Amy Frazier come and tell us how they engage with their community, and then we can have a discussion afterwards about everything we've heard.
Thank you so much, everyone, for inviting me, and to Kaylee especially for her organization. It's been phenomenal. Thank you, really. I'm delighted to be here. Margie Frazier from the Batten Disease Support and Research Association. We're in Columbus, Ohio. Our organization celebrates 30 years this year. And to really celebrate this year, we had our first FDA approval in April. It was very, very exciting. And I'm going to talk a little bit more about why patient engagement mattered so much in that approval. And I'm really wanting to address my comments today to all of the people who are affected by ALD today. We are a family of lysosomal diseases, and I think we share a lot. So let me get started a little bit here. What is Batten? It's another lysosomal storage disease. We don't have great data. I think a lot of lysosomal storage diseases don't have great data, but this is kind of what our scientists are telling us. There are 14 different forms, and they show up in very different phenotypes, and what we're finding is that even when we think we know something about one of the forms, there's tremendous variation in it, and we're finding that with CLN2 that now has Brineura, the FDA-approved BioMarin product. All of these forms of Batten are very life-limiting. With the form that we have a treatment for, kids start to present with an unprovoked seizure at about 2.5 or 3, and left untreated, they will lose their ability to see. They will continue to have horrendous seizures, lose their vision, lose their ability to walk, will be tube fed, and they're gone from us typically about 10 or 11. And another form that we see in the US quite a bit is called CLN3. These kids will lose their vision first and oftentimes are diagnosed by an ophthalmologist and then come to neurology for treatment. They live much longer, actually, and my talk today is dedicated to Jeffrey Monavon. He passed yesterday at 30 years old, and his parents are phenomenal supporters of ours and have just been incredible. We have made the rounds in 30 years. We are connected worldwide with other patient advocacy organizations in Batten disease, which is one of the reasons why we've been successful in getting EMA and FDA approval. We have global family support and research networks all over. I do a lot of traveling. At any one time, we're actively working with about 300 families in the US and Canada. So I have a staff of 4 people, including myself, and we are on the phones all the time. We are very, very busy, but we enjoy what we do and feel like we are making a difference in the quality of life of our patients and their families. I tell my board all the time that the support function in our organization really drives research in many, many, many ways. And one of the ways that really the engagement we do gets done is our yearly conference, which it's a destination for our families. Discussion of the conference begins in March with our families when the schedule comes out. And the conference is in July, and they don't stop talking about it till October or so, wondering when the next one is. We have 450 people every year. I think we'll have more this year because we're going to be in Nashville, or as my friend Frazier Gieselman says, Nash Vegas. We have care for affected kids, and we'll be taking care of about 55 kids. and we take care of their siblings, and we're very proud of the fact that we have a special sibling program, because I think it's very tough to be the sibling in a family with rare disease, because sometimes you don't make it to your soccer game, and sometimes you don't go on vacation because things change rapidly. And so they have 3 days of what we also call free-range hotel, where they are just turned loose, and we have adult sibling leaders who take them on outings, and their parents drop them on a Thursday night, and they don't see them again typically till Sunday because they don't want to. They just want to go off with their people. It's very exciting, and I think it's a national model. We have an incredible amount of online education we do with families. We meet them where they are. A lot of people rue the day that Facebook ever happened, but we have embraced it. We have 900 families from around the world on our closed page, and it's going 24 hours a day. And we do education real time on our Facebook platform with expert panels that jump on. We log them in, and for an hour, our families can ask questions about the topic of the month. And it could be about the keto diet, it could be about seizure control, it could be, last month it was about home modifications. We do them on all kinds of topics, and our families really like them. We de-identify them, and then we post them on our website, so feel free to jump in. We believe it's really important for a human being to answer the phone. I know that sounds old-fashioned. It takes a long time for our families from diagnosis to get the courage to make a phone call to us, because they tell us that when they call, that means it's real. And to face it, that it's real, that your child has this, is very difficult. So we want to be on the phone when they call us. And so our whole office is cross-trained to do intake. Everybody is trained, no matter what, to be able to talk with a family. Sometimes they want to talk to me, but if I'm not there, anybody can answer. And so we want the support to begin immediately and the education to begin immediately. We answer emails from all over the world. We serve primarily US and Canada, but I helped families in 30 countries last year. They're looking for connection. They're looking for physicians. They're looking for technical support. They're looking for entry into trials. They are looking for anybody who can understand them. And it's an honor to help them. I do a lot of business through Facebook Messenger. People are always pinging me, can you help me with A, B, and C? And it's a very powerful medium for us. A lot of our staff are contacted that way too. We believe in warm referrals for local support. And what I mean by that is if somebody's calling me from Salt Lake and they need a referral for something, I don't just give them a phone number. I call the people in Salt Lake and say, can you help this family do A, B, and C? Because our families go through cycle after cycle after cycle of being told no. And I want them to feel like I said yes and that person said yes, so they have 2 wins, and so they're on a roll. I think that's really important for families to feel surrounded and that people are going to be there to help as best they can. Listen, revise, listen. So we're always trying to listen to families to have them tell us what it is they need and how we can change programming. And sorry, Ron, this has taken longer than I thought it would. We believe that these strong family ties drive our research efforts. At our conferences, the goal is to have fun. The goal is not to cry a lot, even though we do cry a lot. The goal is to laugh a lot and bond and have a great time and have a beer or have an ice cream or whatever. But as that conference is going on, we're collecting natural history data, we're collecting blood, we're collecting all kinds of information about families because they're desperate to do anything they can to be helpful, no matter what stage their child is in. We have a robust biobank housed at MGH with the wonderful Sue Kottman, who is a colleague of Florian. She's fantastic and has been a real friend to us. We have a very, very large brain banking system at UCLA, funding in millions for research. We were really pleased about how well we did with patient identification, recruitment, and lack of attrition in our trial. My organization, my team of people, in about 2 weeks, relocated 3 families from across the country into Columbus, Ohio, and they've been there for 3 years. They go every week to Nationwide Children's Hospital to have an infusion. There's an intercerebral port put in their heads. These little kids have this port tapped every 2 weeks with enzyme going into it. They're doing incredibly well, but as you could imagine, this is a real toll that it takes on these families to be away from home. But they continue on, one foot in front of the other, staying overnight in the hospital every 2 weeks. A 7-year-old is the reason we got the approval. The FDA and BioMarin were having difficulties coming to terms with statistics. And BDSRA sent a letter to FDA saying that we want to come see you. We want to talk to you about what we're seeing. And 2 days later, I got an invitation along with BioMarin to come to FDA and tell our story. And 2 weeks later, we were there with 3 families. And one of the families was in the trial, and they brought Maddie. Maddie lives in New Hampshire. He's 7. He had his infusion. He stayed overnight in the hospital. He got on the plane and went to the FDA. He was cranky, but he came. And he had his iPad, and he didn't say much until the end, when we were all sort of having a discussion about what we were seeing in the trial versus what the statistics might have been saying. And Maddie waved and said hi and started talking. And Maddie, at that point, given what we know about natural history and the rapid decline in Batten disease, should have been in a wheelchair on a feeding tube and unable to talk. But he wasn't. And that was big. And so if it hadn't been for the patient voice at that meeting, we wouldn't have had an approval. BioMarin's told us that, and I just heard FDA say that last week in a webinar. It was very exciting. We have a rapid response to research needs. When a company calls me and says, I need a focus group, we can put a focus group together. And the reason we can do that is because we know the patients, they know us, and they trust us. And it takes a lot of time and a lot of energy to have families trusting you. So I want to talk a little bit about what we value, and I promise this is my last slide. It's also my thank you slide. These were, every year at our conference, we make our kids, our siblings stand out in the heat and hold thank you signs so we can send nice notes to people at the end of the year. So they're all out there sweating. We believe that peer support is paramount, but professional support plays a vital role. We want our families relying on that special teacher, that special social worker, that special OT, the PT that advocates for you and knows their business. It's relational, it's not transactional. Everything we do is about building relationships. It's not about what can I get from a patient to make my trial go. It's about how can I invite you into something that's bigger for all of us. We have a radical acceptance of where families are when they engage. Some families are just sort of lockstep forward, let's get this done, boom, 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 boom. They're just ready to go. And then some families are just falling apart. And some families are really mad at me for no reason, because I'm safe. It doesn't matter how they engage, they just engage, and we radically accept wherever they are. Research provides enormous hope in our community, and it's the engine for hope. Cohesion are what make that happen. The way we're able to get families involved. Trust, safety, and outreach. And we say yes as often as we can. If somebody asks us for something, we always try to find a way to say yes. Until we can't. But that's rare. So I want to leave you with this idea that you patients and you parents of patients, you are the loudest and most important voice in the room. Thank you.
Well, Margie, I would like to thank you for inviting me to speak today, especially on a panel titled Learning from Each Other, because Margie and I have talked about this. We know we've been learning from each other for a long time now, and we're both happy that we decided to come at the starting bell this morning and be with you all day, because we've already learned a lot from your community. So least we can do is share our insights with you now. I'd also like to apologize to you in advance. I know, and I appreciated Jean's presentation, because she got emotional in the very first sentence. That's usually what I do, and I didn't want to feel alone. So I know I will get emotional. I know I'll get carried away. I'll get carried away because, and I'm already getting emotional, because like you know, I'm a patient parent, like some of you, and also I've spent the day with you, and I've learned a lot from you already, and I've heard the things that your scientists have said, your family, your parents have said, your patients have said, and it got me all fired up. So I apologize in advance. I'll get carried away and I will get emotional. So I'm going to tell you a little bit about Friedreich's ataxia. First, my name is Ron Bartek. I'm the co-founder with my wife, Rachel, of the Friedreich's Ataxia Research Alliance, or FARA. And I want to tell you a little bit about Friedreich's ataxia, a little bit about FARA. I'll do that through the prism of our family. So back in 1997, when our son, then 11, our son Keith, was diagnosed with Friedreich's ataxia, henceforth called FA for shortness, we sat down on our old Gateway computer and saw plenty of bad news. And that sort of dates it, doesn't it? We were a Gateway holder. So we saw that he was going to get very sick very fast. We knew he would lose a lot of his vision, hearing and speech. He would develop severe scoliosis, or curvature of the spine, requiring implantation of metal rods to get his spine straight and to keep it straight. We knew that he would lose strength and coordination in all four extremities, so he'd most likely be in a wheelchair by his mid-teens. And we knew he had a much increased risk of diabetes. And most compellingly and condemningly, we were pretty certain that he would develop a very serious cardiomyopathy that would take his young life. All that proved true, unfortunately. We saw too that night that there was no treatment. There were precious little research going on. There were no clinical trials being conducted. We sensed, because we thought we were all alone. We'd never talked. We had just that night learned how to. And we knew that there had to be a lot of people out there in this very rare disease that was autosomal recessive, and we knew it was life-shortening. We knew it was relentlessly degenerative, so there was no way to stop it that we knew about at the time, and there's still no way to stop it that we know of. So we figured that there had to be patients like our son and families like ours out there that were unconnected, that they didn't know others exist. They felt alone and helpless and hopeless. And we also saw that the estimate, the statistical estimate, that there was roughly 3,000 to 5,000 American patients and maybe 15,000 worldwide. We called around to a couple of clinicians and found out that there were no clinical efforts going on. We found out too there was no industry involvement. We were hearing constantly, "You're too rare, you're on your own." I bet you've heard a little bit of that. It was wonderful to hear from these gifted companies and scientists today that they are engaged with you and that they're moving forward apace. We did see one piece of good news that night in front of our computer, and that was that the gene had been identified almost a year to the day earlier, in March of 1996. So we figured that if we could start an organization, by the way, that's our son, Keith, in the middle there. And you can see his stance was already at age 4. His stance was wider than his brothers. And his brothers, as it turned out, were Byron is the eldest. He was clear. He had got neither mutation. His younger brother, Stuart, was a carrier like his biological parents. I'm the stepdad. And Keith got both mutations, so is the patient. We decided that if we could start an organization and build up enough awareness and enough resources that with the gene identified, maybe we could make a difference and it wouldn't take forever. So we assembled the 3 leading scientists we found, 7 parents, including ourselves, and 1 adult patient, and established a board of directors and with the mission of marshaling and focusing the resources needed to treat, cure, and now we even say eliminate Friedreich's ataxia. We knew we couldn't do it alone. We knew it was going to take a village. We knew it was going to take everybody, all of our stakeholders working together. And I sense that's the same spirit in this group. We knew in doing that, we would have to work with our patients and families, the academic investigators, the industry partners, the government partners, and very importantly, other advocacy organizations. We were the new kid on the block, and we were the only organization in the United States that was completely devoted to funding research in our rare disease. There were others, like the Muscular Dystrophy Association, that had about 42 different rare diseases. We were one of them. National Ataxia Foundation had 32 ataxias. We were one of them, but we were the only one devoted entirely to supporting this rare disease research. We knew in all that collaboration that the kinds of trust and community that Margie had just described was going to be the rock. It was going to be the foundation of everything we were able to do. We would build on that. We knew that our patients and their families would provide the fuel and the driving force behind advancing the research. Another way to say that is the patient engagement is in all we do, it is all we do. And when I say patient in this case, talking about the patient family, and the patient advocate. All of us in this room would qualify in that definition. So how do those patients, family members, and advocates help in this effort? One way is in fundraising. Grassroots fundraising in our organization accounts for 1/3 to 1/2 of the $6 million plus we raise each year. Almost all of that devoted to the research. And is the basis of the relationships we built with the other advocacy organizations. First thing we did as we were founded was I flew to Tucson to talk with the Muscular Dystrophy Association Board of Directors, flew to Minneapolis to talk to the National Ataxia Foundation Board of Directors, flew to Australia to talk with a patient group there, to Europe to talk with patient groups there. And we just wouldn't take no for an answer. It was going to be collaboration, collaboration, collaboration. We knew that none of those groups, including ourselves, could do it alone. And we wanted to build the kind of cohesiveness and community that would provide the foundation on which we could get it done. They also help us in advocacy. The patients and their families provide the very foundation and basis for all the advocacy work that we do. This woman right here asked the question, although she told me she knew the answer, asking about how we advocate. And I just wanted to say that our organization itself, and in collaboration with other umbrella organizations like the National Organization for Rare Disorders, the Alliance for a Stronger FDA, Research America, Every Life Foundation, Research, Global Genes, and the Genetic Alliance, we combine efforts with all those organizations. We go to the Hill a lot. And a lot of you probably have been involved in Rare Disease Week. We heard one reference to that. It's coming up again, as it is every year in February, the last week of February, where all of us go to the Hill and advocate, yes, for common issues, but also for individual issues. You could go up there with the whole group and advocate for ALD. So it's just a wonderful opportunity to get everybody together. We always advocate every year for more resources for the NIH, more resources for the FDA. We know that everybody in this room, everybody in all rare diseases needs those two agencies every day, all year round. So we always advocate for their resources. We also advocate for specific congressional pieces of legislation, like last year's wonderful passage of the 21st Century Cures Act, which authorized considerably more funding for both the NIH and FDA. And right now we're deeply engaged in something maybe you know about, and that's protecting the wonderful incentives that our FDA colleague told you about this morning for, based on the Orphan Drug Act of 1983. He talked a lot about the 7 years of exclusivity, but the 50% tax credit. It's called the Orphan Drug Tax Credit, whereas a company like the several that have just spoken with you, if they're developing a drug for a therapy for ALD, which is a rare disease, and they go to the FDA and they qualify for and receive orphan drug designation, then they can get a return of all their investment in that R&D for that specific drug, for that specific indication, in the form of tax credits. So they would get returned to them 50% of their R&D costs. That provision is completely under stress right now in the Congress. The House of Representatives passed its tax reform act yesterday. It would completely repeal the orphan drug tax credit. The Senate has a draft out that it will consider over the next 2 weeks. It would probably cut it in half. We've been spending a lot of our time going to the Hill, trying to talk some sense, into these legislators, saying you don't want your 30 million rare disease patients in the country paying with their lives for the tax savings that you're trying to gain. And so you might consider teaming up. I don't know if you guys are members of one of those organizations. We're all in it together, NORD and Every Life Foundation and so forth. They're all lobbying for that. So the patients help tremendously with that. The patients also help themselves, help each other with communications, education, encouragement, and inspiration. This patient-to-patient communication. We've got a set of now 50 ambassadors, patients themselves, who communicate on all the social media with each other, keeping each other's spirits up, informing people, their colleagues, about clinical trials that they informing them about advocacy efforts that are ongoing. It's a wonderful program. You'll hear tomorrow afternoon from our dear colleague, Kyle Bryant, who is our ambassador. He runs the ambassador program, and it's just really a powerful way that our patients get involved in our everyday operation. And so they're constantly personally inspiring each other. They're constantly personally inspiring all of our other partners. They do grand rounds, as our FARA management team does, grand rounds at industry partners, grand rounds in the government. And we've heard from any number of industry partners and any number of labs that these encounters have changed their lives. And they now know what they're doing, why they're doing it, and for whom they're doing it. So usually there's not a dry eye in the house. And this is, we've done it with big companies and small. We've done it in government labs. We've done it in government institutes at the NIH, and literally not a dry eye in the house. And they go right back to their labs and get to work. These same patients, of course, provide all the preclinical tools. They're the ones that are, giving their blood samples, their urine samples, their cheek swabs, their skin biopsies. The skin biopsies become fibroblasts that then educate the development of iPS cells, induced pluripotent stem cells, that we're using for test beds. And probably most importantly, these are the patients that enroll in our patient registry. We've got about 3,000 patients enrolled in our patient registry. They're the ones that go to our clinics and get examined. Those examination data populate our natural history database that now has about 1,000 patients in our natural history database, and they've gone for these annual examinations for anywhere between 1 and 13 years. So we have all those natural history data across 13 years with 1,000 patients. And that is a powerful tool, combined especially with the with our clinical network. We've got now 9 sites in the US, 1 in Canada, 1 in Australia, 1 in Brazil, and we collaborate fully with about an equal number of sites in Europe. And they're all doing clinical research on the same protocols. They're doing basic research, so that combination of the patient registry, which is a demographic registry that we use for recruitment, and the natural history database and the clinical network, those are the powerful magnets that will bring industry partners to you. Good illustration of that is when we got started in 1998, when we founded the organization, we had no industry partners. Again, too rare, you're on your own. We now have a little over 30 industry partners, 9 of which are funding clinical trials for us. We have 9 trials going on right now. 2 of them are phase 3 clinical trials. We've got 4 companies and 2 universities rapidly and aggressively advancing gene therapy that should be in clinical trials beginning by the end of next year. We should have a protein replacement strategy in clinical trial next year, a histone deacetylase enzyme inhibitor program by the middle of next year. So the cavalry has mounted, and they're all mounting because of these patients and their engagement and enabling these natural history studies, the patient registry. And we use the patient registry to recruit trials in record time. We recruited a 60-patient, 3-site trial across the US, phase 3 clinical trial in 2 hours and 43 minutes. And we're going to get faster. So, that's why we have 30 partners now. They know we will save them a lot of time and money, and they can use our natural history study, as you heard several of them say, when asked, what's the most important thing we can do for you, as Ali asked them, most of them said data. And, they know they can come to us now, as they often do, if they've got even before they've got a clinical candidate, but certainly when they do have a clinical candidate, they will spend a day with us and our key opinion leaders and our staff and our best clinicians and say, what does your natural history data show you about what kind of study we should have? How should we design the protocol? Which patient population, subpopulation should we use? Which sites should we use? What procedures should we use? What endpoints and biomarkers can we mine from your natural history data? So they will sit there for a full day taking notes. They'll go back to their buildings and draft a protocol, send it to us to vet, to say, to give recommendations about. So that's how significant they think the help, the real answer to Ali's question, I think, for us is we can help by providing patient registry, natural history database that's richly populated, and a clinical network that includes clinicians and nurses that know our patients, know our disease, and are conducting most of our clinical trials. To summarize too, what Margie said, the voice of the patient has greatly enhanced all of those relationships. Which leads us to, you heard from a couple of the industry partners a little while ago how important their relationship is with the FDA. And there's probably nothing more important to our industry partners than the relationship we have with the FDA and the relationships that we help them build with the FDA. And a great illustration of that recently was we had our what's called externally led patient-focused drug development meeting nearby in Silver Spring, Maryland, about a 5-minute drive from the FDA. We assembled 150 patients live in the room and about 200 others on the webcast and participating live. And they gave, it was called the result is a report that's shown right there, right somewhere there, called The Voice of the Patient. And so for most of a day, we had patients and caregivers speaking really to the FDA. There were more than 20 FDA people in the room all day. There were 25 pharma partners. I gave opening remarks. Jen Farmer, our executive director, gave opening remarks. Then the FDA's opening remarks were given by Dr. Wilson Bryan, who is the director of the Office of Advanced Therapies, Tissue and Advanced Therapies. That's the review office that will review all the gene therapy proposals. They're the ones that reviewed your gene therapy proposal. So he had hosted a meeting with our group, our 4 companies and 2 universities doing gene therapy in January, and he gave the opening remarks at our patient-focused drug development meeting on June 2nd, told the story of how one of our patients, when he was a doctor in Texas, had changed his life. So, and then if you fast forward to 10 days after this meeting, I'm on the phone with one of our industry partners and the review division on the other side of the FDA house at CDER, the Center for Drug Evaluation and Research, the review division that does all of our small molecules, not the molecules. His team had been there all day with us as well. And so I'm on the phone. I'm asked to kick off the meeting. I thank them for being at the June 2nd meeting and listening to our patient's voice. And I summarized the patient's voice for him. He stopped me almost. He said, "Mr. Bartek, you've got no reason to be thanking us. We should be thanking you for giving us the opportunity to hear your patient's voice. We heard it. We heard it loud and clear." He said, "We know what an emotional issue this is for you. We want you to know June 2nd made it an emotional issue for us. It had and not only that, but it had a powerful scientific impact on us." He said that combination of emotional and scientific impact has already changed the way we will do our business with your disease group. I mean, what more could you hear from your regulator? They want it to work for all of us. They are really all in. And he went on to say that, we had some doubts about your endpoints, that these classic neurological scales were not, we were concerned that they did not sufficiently reflect that the patients were actually feeling and functioning better. that they were able to perform their activities of daily living better, that it was having a positive impact on their quality of life. He said, and then after that June 2nd meeting, he said, okay, here's what we learned on June 2nd. We learned that you can go ahead and use your classic endpoints. You know, we have a fairly well-developed Friedreich's ataxia rating scale, so neurological exam and some of the performance measures you guys have talked about today. 25-foot time walk, pegboard test that we borrowed from the Multiple Sclerosis Society, and a visual acuity test. So go ahead and use that. That's fine for your primary endpoint, but give us additional evidence that the patient is feeling and functioning better. And especially since most of these drugs you're going to try are not going to give you a cure. They're going to give you incremental improvement, right? Which has been the case so far. So if you're showing a modest improvement, incremental improvement in these classic scales, give us more evidence from the patient that they're feeling and functioning better, able to perform their activity of daily living better. What they're really saying, and then the CEO of the company intervened and said, well, does it have to be against a validated outcome measure? And the director laughed out loud. He said, no, I don't even know what validated means. Just give us more evidence that the patient's feeling and functioning better. This is so phenomenal in terms of news. I know all these drug company people in the room and your clinicians will know the reason that what they're really saying is, yeah, give us your primary endpoint data, but give us another reason to say yes. Give us another reason that, okay, we may be seeing an incremental improvement, but the patient's saying, I feel and function better. So this was just a monumental improvement in our community. So now, so there will be an exam on every word in this slide. Some of you have seen this before. This is a slide we helped the Clinical Trials Transformation Initiative develop a couple years ago. It really just shows very fundamentally that with the patient engagement that we've been talking about, we can actually affect the action throughout the R&D spectrum, throughout the R&D continuum, from basic discovery science on the left, all the way to the approved therapy and post-market surveillance at the end, and all the way, you know, your interest in clinical trial readiness right down the middle there in phase 1 and phase 2. Patients are engaged every step along the way and advocates are involved every step along the way. And you know, it's marvelous. And plus that, the NIH and the FDA know that, and they hunger for it. I mean, that's the way they want it to go. Last slides. Come here. What am I doing wrong? So that's part of the proof of the pudding, that we're engaged at every step along the continuum. Here's another proof of the pudding. Here's the FARA, Friedreich's ataxia treatment pipeline. When we founded the organization in 1998, we had only 3 mechanisms that we could find that had been explored. None of them were in clinical trial just yet, but 3 potential treatment approaches. And this is our pipeline today. So you can see 9 compounds in clinical trial, and some of them are some later or earlier stage compounds are really profoundly beneficial potentially. So we're really thrilled with this, and it's another tribute to the fact that patients are engaged throughout the process, and the drug companies are coming to our side now and helping us develop these things. So I'm not going to go through this. This is one case example of how a company that's just entered a phase 3 clinical trial, how we exercised every one of those steps with them using our natural history studies and our patient registry, and giving them advice all along the way in trial design and so forth, recruiting their trial very quickly. and led to their chief medical officer saying the following: investigators and coordinators in the FA, our network, are incredibly responsive and professional. With their partnership and fair leadership, we're able to identify interested investigators and sites, develop a phase 2/3 protocol, obtain regulatory and ethics committee approvals, and launch a study in approximately 6 months. Once the study opened for screening, the first cohort of patients was identified, recruited, screened, and enrolled in less than 2 months. So that's how we roll, and that's our experience. Again, thank you very much for having us.
Great. Dean, am I on? So I couldn't have asked for two better advocates to learn from. So consider yourselves now official mentors of ALD Connect. I model to follow that sort of learning from each other, and whether it's big sister or big brother, I think that. But Jean, are you here? Where are you? Oh, right there. I thought maybe we can open this up for questions, and you've heard different presentations, and I wanted to get some reactions and questions. I want ALD Connect to have its Hammerfest, I guess, is how I look at this. I guess Margie, listening to Jean and thinking where we are, and what would you say projecting 10 years forward based on the Batten's experience? Can you reflect well, what you think lies in our future and what we should do?
I think you're probably closer than that. I was surprised at the rocket speed that BioMarin took on our project. I know there's more in the pipeline in other companies, and we've got trials opening this year for other forms. And just putting one foot in front of the other, collecting your data, making sure that you've got natural history. And it doesn't have to be perfect. I think we always think that it has to be, it's just like Ron just said about FDA, that it didn't have to be validated for the quality of life. They just wanted some reason to have other information, and that was our experience as well. Just keep at it. Mm-hmm. The heartbeat of our BioMarin project was a family. They're the ones who went to BioMarin. They're the ones who push, that they understood that they weren't going to do it by themselves, that they were going to need the patient group to push with them. And they needed an umbrella group like ours to be able to pull patients together for a variety of purposes and attend advisory board meetings and fill in the blanks, because that's a big endeavor. I think your future's bright. And, and the other thing I wanted to say is that I've learned a whole lot more today than you probably have learned from me. I think your model, there's a lot of things that our community needs to learn from you in terms of how you've organized industry partners and medical and scientific communities for a common goal. I think it's easy, actually, for rare disease groups to splinter and for their researchers to splinter, and it's not easy to bring people together and keep them together. It's a constant sort of bringing people to the party, and so I think you do it really well, and it's important.
And I'm like Margie, I'd like to reinforce the notion that natural history is so vital. And you heard it from your industry colleagues, you heard it from others today. In my view, there's nothing more important than natural history data as a tool to bring industry partners to your side. I'd also like to congratulate you for having both the NIH and the FDA here today. I think that was just really important, and want to reinforce especially what Jill Morris said about the importance of working with our program director at NINDS, who is Jill, and that function is so vitally important to everything you're trying to accomplish. And I fully encourage you to get more and more involved with Jill, to get involved in that National Advisory Council she mentioned. It meets 3 times a year, a full day. 3/4 of that day is an open session that you could all go to, and it's so cool to be there in that room with all the program directors, the whole institute management, the director and all of his staff, and get to know these people, get to communicate with, and to communicate to them how important they are to you, and to learn about the new programs, to learn about the mechanisms, the grant mechanisms that you can enjoy the benefits from, and that you as an organization can help support your academic investigators and your companies by sending letters of support to Jill. And you heard her say, and this has happened to us, that there are times when they will reach out to you because they've got a grant proposal that maybe is right above the pay line, is not going to get funded. To say, they've called us a couple of times to say, this one, isn't going to quite make it unless you tell me that this is really important to your organization, really important to your disease community, and it's worked. And so it's really a powerful relationship. This town is all about relationships, and that is, I would say, the relationship with NINDS program director and that whole institute, and your relationship with the Food and Drug Administration are the two. I'd put Congress in 3rd place. Very important, but well, I'll just go on to say NIH and the FDA are absolutely essential.
Sort of harking back to what Margie said about things are relational, not transactional.
Yeah,
that probably comes back to this relationship as well. I want to just connect to what you said about natural history, since we've been discussing this as a board as well, how to best go about getting natural history and what the nature of that natural history should be.
Okay.
Because we have different platforms, different layers. We have, the patient voice. We have a patient portal. We have, physicians who are very busy who we want to have them enter data in a standardized way. And how do we harness all this and do it in a way that makes most sense? And I was thinking of what Margie said about, well, we have, advocacy meetings and people come and we gather natural history as they're there. How does all this fit together? Ron, help me make sense.
Okay. We took a different approach. Our approach was, and it was almost blind. We decided way back in, I think it was 2003 or something like that, that what we really needed in order to conduct our first clinical trial was outcome measures, endpoints, and to measure success or failure. And that was true. So we said, well, how are we going to get that? And the decision we made right away was, let's get a small number of patients. We chose 14 patients from this catchment area, the DC area, and we scheduled a full day at the NIH. NINDS was hosting us. We gathered 10 clinicians from our community. We had each of them examine each of those 14 patients separately. Each patient went around to 10 stations and had this we chose a classic neurological exam as the first step, with all the things you've all been through. And then we picked a set of what we call functional measures. And we borrowed 3 of them from the MS scales. 25-foot time walk, a 9-hole pegboard test, and a low contrast visual acuity test. And there was a speech test, because speech is a real issue in our disease as well. And so each patient went around 10 times and had all those things conducted. We did that again 18 months later without intervention. There were no drug therapies or anything. And we got 12 of those same 14 patients and all 10 of the same clinicians. They did all of those same exams. They looked at all those data afterwards and they said the most sensitive measures to change across 18 months of progression were the following. That became our Friedreich's ataxia rating scale and our functional measures. And then we funded, we chose 5 sites initially, 5 clinical sites, 4 in the US, 1 in Australia. And we started encouraging patients to go in and get that examination. And we thought we were just looking for endpoints, what we call ataxia scales. And we spent $32,000 was all on 2 towers to house those data. And then, wow, it was really amazing. We went to University of Rochester Institute of Technology, a student program in IT. And we had them, as a capstone exercise, design the data templates, 60 data templates to house those data. And put those, and then, so everybody had the same templates. They were entering data against the same exams in the same template. So for uniformity, and all those data collected at the Children's Hospital of Philadelphia. And that went on for 5 years. That we saw right away, this is looking a lot like natural history data. And we started adding exams, and adding a hearing exam, adding heart measures and stuff. So anyway, that's the way it got built. Now it's all housed up at the University of Rochester in Rochester, New York, against a really sophisticated backbone.
Ron, quick question. So how do you encourage clinicians? I understand that patients definitely have their incentives to donate data through exams and regular to clinics.
Right.
How do you encourage, I don't know, financially or somewhat other
Well,
ways?
we have a high
triathlons.
Yeah.
And so.
You want to take that first?
We've just been incredibly fortunate.
Yeah.
Actually, Alfred Kohlschutter, he's just a gem. Started ours, the DEM Child database, and now we have feeders from US, South America, the UK, all over Europe. It's a beautiful thing. And it needs work. Mm. And FDA will tell you that. It needs work. That's why they were discussing statistics for so long with BioMarin. And so I'm a social worker by training, and we are trained to find strengths in things. That's how we assess people. You start with strengths. And so I say you start with what you've got, you assess the strength of it, and you build on it. And I think your model for ALD Connect is brilliant because you are devoted to bringing people together to connect. That's the beauty of it. And so I think, we have had whole conferences just on outcomes related to our natural history database to figure out what next steps are, what refinements need to be made. And truthfully, I think, we heard this from FDA, we need much more on quality of life and those measures. We're working on those. We have actually 2 rating scales in our orbit. Those need to be unified and made stronger and validated. Mm-hmm. And we're in the process of that, and we're really happy to say that if you do a unified Batten rating scale in Hamburg and you do one in Rochester, that data matches about 96%. Yeah. That's a beautiful moment when you're talking about different languages, rating people. That's great. We have some incredibly skilled people. It doesn't happen overnight. I also think that there's nothing like a clinical trial to teach you how good your data is either. And we're very indebted to the R&D partners in industry helping us as well, because they've worked on other trials and they know really where we should be going, and the FDA as well. So it's a partnership, and we have to look at it that way with government, industry, and nonprofits.
Yeah, I'll just echo that. It's what she said. You know, the beauty of your model, pulling this everybody together, is exactly the way we've been able to do it. We like to say that acting alone, there's very little we can accomplish, while acting together, there's very little we will not accomplish. And you take that natural history study, for example. We got it started at 5 clinics, and we convinced the Muscular Dystrophy Association to pay for the whole thing the first year. It was $400,000 the first year. They paid for the whole thing. The second year, they paid 2/3 of it. The third year, they paid half of it. We added a 4th year, and we split it with them that 4th year. And so we were paying percentage of effort on the part of the PI, and we were paying pretty much for the full coordinating staff. And then for a while, we kept it on that basis, percentage of effort for the PI and pay for the nurse coordinator. We've now shifted to fee for service. So they get paid a certain fee for every patient data they enter. Back to a question I think that was asked earlier, each clinician, I think you and I were talking about this, Alex. Each clinician has access to the data that clinician entered into the overall database. Only the overall PI has access to all of it. We don't give our industry partners access to any raw data. We don't want it being abused or misused. We give them full access to analyze data. And we've got a really good team at University of Rochester to help our PIs analyze the data. And then we sit down with our industry partners and use those data analyses to help instruct their clinical trial design.
Thank you, Ron. So thanks for sharing. And usually they say that with my accent I'm too direct. So let me ask you a very direct question. So out of those 6 million plus,
yes,
how much are you spending on average annually on the natural history study? 1,000 patients, annual data entry, annual exam.
It's minimal. I'd have
Order
to look at that, Alex.
Is it $500,000? Is it
that would not be a
$5 million?
bad estimate. I'd say 400,000 to 500,000, maybe something along, maybe 600,000 in that ballpark.
All right, thank you.
Yeah.
Great. This was very instructive. Let's go into a short break and we'll go into our last session. Thanks so much again.
Thank you. Thank you.